agar with agarose. Otherwise, that gel results will look pretty gross.
Agar and agarose are both made up of long chains of sugar or polysaccharides that come from red algae or seaweed, and which we can use to make gels to, say, separate DNA fragments by size or fill with bacterial foods or media and allow bacteria to grow on them in petri dishes. So, what's the difference? Well, agar is actually a mixture of agarose and another type of sugar chain called agaropectin. Agarose are these long chains of these repeating sugar units. We'll go more into the details, but these units are neutral. And so what's going to happen is that they're not going to interact strongly with charged molecules. And this is going to make them useful when we want to do DNA gel electrophoresis, where we're separating DNA, which has this negative charge based on electricity being motivated by charge. And if we had a charged gel, well, this would cause problems because our DNA would react with it. And so by using this agarose, which is this neutral form, we're then able to separate DNA without the DNA actually interacting with the gel. We can instead just use the gel as a mesh for separating things. But in order to actually get that agarose, we have to purify it out of agar. And because agar also contains agaropectin, which has modifications, including modifications like sulfur, which are going to be negatively charged. And if you imagine what would happen if you tried running DNA through an agar gel, it would kind of get repelled by those groups and it would make your gel run really weird. But we don't really care about that charge and variability if all we want to do is make a gel. And agaropectin is fine for being in there if we want to make a gel. And so we'll talk more about how this gelation process occurs. But basically, both agar and agarose can kind of knit themselves up into these reversible gels that make them really useful. And these gels are going to be able to be used to fill them with bacterial media, so this bacterial food, and then we can basically just grow bacteria on them. And we can include antibiotics in that food if we want to do things like use antibiotics as selectable markers. So say we put a circular piece of DNA called a plasmid into bacteria, and we have an antibiotic resistance gene in that plasmid. Then only cells that take in the plasmid will be able to grow in the presence of that antibiotic, and they'll grow as these like colonies, these gloopy dots, which then tell us, okay, well, this colony has our plasmid of interest. And so for this, it doesn't matter if those chains are going to be charged or not. And so we can use agar for this. Agar can also be used kind of like by food makers and things like this because of that whole gelation property. And sometimes the modifications can actually be helpful because they can allow the gel to, say, hold more moisture, which can make affect the taste and the texture of foods and things like this. This idea of the modifications affecting moisture brings us to, okay, well, how do these gels actually form? What's really cool about these gels, or one of the things that's cool about them, is that unlike with polyacrylamide gels, so things like if you're running an SDS page to separate proteins by size, or you're running a TBE urea page to separate small RNA molecules, those gels are going to be chemically cross-linked. So you actually have permanent interactions, permanent links between the chains, and so they're non-reversible. But in the case of our agar and our agarose gels, what we have is our chains are more kind of just like a knit scarf. The chains are going to be held together by attractions, not permanent bonds. A little more detail. So for the actual more background, go check out other posts. But if you look at each of those sugar units inside of your agarose chains and also inside of your agaropectin, and we'll get more into the details of what these are made up of in a minute. But you can see a lot of these OH groups, these oxygens connected to a hydrogen. And this offers up the possibility for something called hydrogen bonding. Oxygen and hydrogen and all those other atoms are linked together within the molecule. So within this, all these where you see a line, that's representing a covalent bond. And a covalent bond is one of those strong bonds. And the strong bonds are formed by sharing of pairs of electrons. And so each of these oxygens and carbons and hydrogens are made up of smaller parts, these subatomic particles. We have the positively charged protons and the neutral neutrons hanging out in the nucleus, and they're surrounded by these clouds of negatively charged electrons. And it's these electrons that these atoms are going to share pairs of to form these covalent bonds. Now, they don't always share fairly, and this can lead to an imbalance of partial charge, even if your molecule is neutral overall. So if we look at the repeating units in agarose, this is called agarobiose, this disaccharide, this 2-sugar unit. We'll look more at this in a second. But you can see that basically these have these OH groups that are going to then be, the oxygen is going to be hogging the electrons it's sharing with this hydrogen, and it's going to make this oxygen partly negative and this hydrogen partly positive. And if you were to then go and look on another one of these units, you would see, okay, well, this partially negatively charged oxygen can now hang out with this partially positively charged hydrogen, and you get an attraction we call a hydrogen bond. And these are happening even though these units are neutral overall. So even though the benefit of having this agarose chain is that it's electrically neutral, or that's one of the benefits, it's still able to form these partial charge-based attractions called hydrogen bonds. And because you have these long chains where you have all of these OH groups, you're able to form lots and lots and lots of these hydrogen bonds. And you're able to, when you do this, you're kind of able to knit up a gel that can then hold water. And you can influence how meshy you want the gel to be by influencing how much of the agarose you want. And this gel can also be formed in the presence of the agaropectin. And with agaropectin, you might get some slightly different interactions because you have more modifications. But both of these can form these gels with this really cool property called thermal hysteresis. Basically, because those those bonds are basically attractions, they're not permanent bonds. There's no electron sharing. There's no actual like pairs of electrons being shared between those chains. This makes it so that that kind of like knitting up, the forming of the gel, the gelation is going to be reversible. And so what's really, really cool is that this reversibility actually has this cool property called thermal hysteresis. This means that it melts at a higher temperature than it gels. So in order to actually get this gel to form in the first place, what we do is we take a powder. And so this is agarose, and if you you can also get agar or agar already mixed in with the components of the LB media. So LB is this common bacterial food we use. We actually buy this mixture of the components of LB mixed with agar, and then we just dissolve that in water. Now, when we dissolve one of these sugar powders in water, what's going to happen is that those individual sugar chains are kind of going to get coated in water. And this is going to kind of get those to free up a little. And when you add energy, you add heat, which you can do either like in an autoclave or in a microwave. Now those chains that were kind of stuck together in the solid form, they were kind of just clumped together. Now they're going to be able to find their optimal partners for that hydrogen bonding. They're going to be all wiggling around, but then when you cool it down, they're going to kind of have to settle down until they find the optimal hydrogen bonding partners. And when they do so, they create this mesh. But the temperature at which they go and they form that gel, this is the gelation temperature. And it's going to be lower than the temperature at which that gel will then melt again. So when you take that dissolved mixture, you have a little bit of wiggle room before it's going to solidify. But then once it solidifies, you're able to run a gel that's going to be stable at a higher temperature than the temperature at which the gel solidified. And this allows us then to do things like run gels without having to worry about our gel melting. But if we then want the gel to melt, we can get it higher again, such as if we didn't pour that agar in time, so we can actually make it ahead of time and then microwave it when we're ready. Or if we want to melt the gel to purify the band of DNA out of it. So this property of reversibility is going to come in super duper handy. And that's a property of both the agarose and the agar. And so let's talk a little bit more about the chemical structure of these two. As I mentioned, agarose is made up of these disaccharides or these two sugar units called agarobios. So when it comes to glycobiology or the biology of sugars, we can classify different sugars based on how many of individual units they have. And so we saw in the case of this agarobios, we kind of have these two units, and these individual units are called monosaccharides with mono one saccharide sugar. And so they have these one sugar. And in this case, the sugar that we have, there's actually two different ones that are linked together. So there's beta D-galactose and 3,6-anhydro alpha L-galactose. Don't get too scared away by all the naming. Galactose is one of the 9 main monosaccharides, at least that our bodies use. And basically, when it comes to that beta and alpha, this is just referring to the direction that these sort of arms are linking off of the sugar from. So it's a stereochemistry thing, or it refers to the orientation in space, which therefore affects how the sugar chains can link together and kind of the shape that those chains will make. In the case of this agarose, we have this through a beta 1,4 linkage. So you get a linkage something like this. If we look at this anhydro thing, basically an without hydro, water. And so we can see that we're basically losing a water molecule in order to link these two together. And so we get this kind of bridge over the molecule to get this 3,6-anhydro alpha L-galactose. Where the 3 and the 6, this is just referring to the carbon numbering. So that's all this is. So we link those together to get this agarobios, which is a disaccharide. So it has two sugar molecules linked together. Note that a disaccharide you're more familiar with probably is lactose, which is going to be a combination of glucose and galactose, rather than galactose and this altered galactose. But in agarose, we have this agarobios, and then lots of these link together to give you agarose. So there's typically about 400 or so. So you have about 800 of these galactose units per agarose chain. And remember that these chains are going to be electrically neutral, but they're going to have that partial charge that's going to allow them to form these hydrogen bonds. So you get the best of both worlds where you're able to get this gel formed, but this gel is going to be neutral. So it's going to be good for separating molecules like DNA. And these chains are going to be fairly consistent in their length and things. So you're able to create a fairly consistent mesh, although not nearly as consistent as the mesh you would get with the polyacrylamide gel, and not as fine, and not as like sturdy too. So one of the main places you'll probably see agarose used is going to be in these agarose gel electrophoresis. But there are also places it's being used where you might not even realize it, including with size exclusion chromatography, as well as like desalting columns. That same neutrality that makes it good for separating DNA also makes it good for separating other molecules when we don't want those molecules to actually be interacting with the matrix or with the gel, which is the thing that we're using to separate. And so this comes into play with this technique called size exclusion chromatography or gel filtration, where we have these little beads, and we can have these beads made up of agarose. And we call it gel filtration because basically these are little tiny gels. You can think of them kind of like little agarose gels, but in these bead forms. And then you have these columns filled with beads, and you can have big columns and little columns. And basically, these beads have different meshing niches. And the big molecules, they can't fit into the smaller tunnels, those smaller holes in the mesh. And so they get to go around the beads, and therefore they have a shorter path. Whereas the smaller molecules, they have to go through all those pores in the beads, and therefore they're going to travel slowly and they're going to come out later. If you have a really big difference in size between the molecules and you have really tiny pores, you can actually use this to kind of trap salts and other small things inside the beads to desalt your solution. So get the proteins and the big things that can't fit in those beads to go around them, and you can get the little things to get stuck in them. Or you can use bigger pores to kind of separate molecules, separate proteins based on their size, but still get them all kind of to come out. So you run it longer so that everything comes out, but they're going to come out at different distances. We're relying on the fact that the distance that they come out at is going to be dependent on the size of the molecules. But if those molecules are interacting with the beads themselves, well, this would cause a problem because now it wouldn't be directly related to the size. And so we want to use something that's really blah. And so the same blahness of agarose comes into play, makes it really handy for separating these molecules with size exclusion. And so a lot of those beads that you see are going to be made up of agarose. Although because agarose isn't that sturdy, they often have some of those more permanent modifications to kind of cross-link those different sugar chains together permanently, so you get a sturdier mixture. So that was agarose. Now let's talk about agar. So agar is about 2/3 agarose and about 1/3 agaropectin. And agarose is going to actually be made by purifying out the agarose component of agar. So before we go into the details, you can already see kind of one of the benefits of agar, if you don't care about them being modified, is that you don't have to go through that extra purification process. So it's going to be cheaper. In terms of the modifications, these modifications come in a variety of different flavors, and they're going to be kind of randomly distributed. So they're not consistent. Every batch of agar that you get is going to be a sort of different mixture. And so there's going to be more variability. This variability can come from a couple different places. It can come from the length of the chains. So remember that in agarose, you had those chains that were about 400 agrobios units, or about 800 of those individual sugar units. With agaropectin, the chains are typically shorter, but it's not like they all have a set length, and they can be very variable. What can also be variable is going to be the modifications. So there's different modifications, including methyl groups, so these CH3, sulfur groups, which we've talked about a lot because they're going to be those negatively charged groups. But there are also different modifications you can get, like pyruvic acid and things like this. And again, these are going to be kind of scattered throughout. So on average, you might have every 10th galactose be attached to a sulfate through an O-sulfate linkage. So that's basically attached through one of the oxygens. But that's on average, and the modifications are really scattered throughout. So it's a mixed bag that you're going to get. What's good, though, is that this you can still get this mesh form, and these bacteria don't care if there's going to be these modifications. And what's great too is that the bacteria, at least most of them, aren't able to digest the agar, and so they're not going to eat the land that they're on, only going to eat the food that we put in there. And if we put antibiotics in the food, well, now we can use these plates for antibiotic-based selection. And both of these, remember, are going to have that property of thermal hysteresis, where we can boil the sugar mixture in water, or in our buffer more commonly, or in the bacteria food. And then it's going to kind of, when it cools, it's going to knit itself up. It's going to form those hydrogen bonds that form this mesh. But the mesh, at the temperature that it forms, it's still going to be stable because you kind of already made all those connections. You're going to have to get it to a higher temperature in order to get it to melt back again. So we have some wiggle room where it's okay if our gel gets a little warm during our run, or when we stick the plates in the incubator, the gel is not going to melt until we get to a much higher temperature. And so this makes these gels really, really useful in the lab. So remember that agarose is one component of agar, and it's this more neutral and consistent component that makes it great for separating molecules by size, such as in your DNA gel electrophoresis or in your size exclusion chromatography, where we don't want the molecules to interact with the agarose because we want them to basically be separating based on just their size, and we can get them to separate based on their size by kind of using these gel mesh as a sieve, and we can influence how meshy it is by modifying how much of the sugar we have in there. Agar is going to be about 2/3 agarose, but also about 1/3 of a mixture of sugars that we just refer to as agaropectin. And so these sugars are typically shorter in length, and they've got a lot of modifications, including things like methyl groups and sulfur groups, which can be charged. And this charge is going to make them really bad if you want to separate molecules based on just their size without having to worry about those molecules interacting with the thing you're using to separate them. But if you don't care about the molecules interacting with them, if you don't care about that sort of thing, then you can still make a really great gel out of them. And so you can make this nice home for bacteria. You fill it up with this bacterial food, this media, and you make what I like to refer to as bacterial pancakes, but these like LB-agar petri dishes. And you can stick antibiotics in there if you want to select for bacteria and have an antibiotic resistance gene. Makes them really, really helpful for cloning and things like this. And because we don't have to worry about purifying out that agarose, we're able to get this agar cheaper. So that is the difference between agar and agarose. So remember that agarose is the more purified form. It just has that long neutral component. We use it for gel electrophoresis and size exclusion chromatography resins. And agar is going to be a mixture of agarose and agaropectin. So it has more of this variability, including modifications that can be charged and would make it bad for running a gel, but good for making gels that we can fill with bacteria food and have them grow on.